Doug Kaufmann is the host of a popular TV show, Know the Cause. He became interested in the subject of fungus as it pertains to disease while working in the area of food allergy. Doug was a U.S. Navy medical corpsman attached to the 7th Marine Division in 1970. Upon coming home from Vietnam in 1971, he had strange symptoms that until 1980 he thought were related to food allergies. A research paper entitled Anti-Genetically Intact Food Macromolecules Exiting the Gut Lumen connected the terrain of the intestine to food allergies. After studying the reason that foods were able to leak through the intestine, Doug saw the role of fungus in provoking both symptoms and disease. He has authored 9 books on the subject of fungus and ill health, thus the stimulus for requesting his expertise at this conference on fungal parasites dental conundrum. His session will be embellished by case studies presented by Dr. Christine Salter. She is an MD and a DC in the St. Louis area and leads the Center for Vibrant Health and Wellness that offers a unique, integrative, holistic medical program to achieve optimal health. I'm certain that Chaplain Paul Johnson, who is the bookends of this conference with spirituality, would appreciate the mission statement from the center. It recognizes the sovereign God as the creator of the human body, as well as the ultimate physician. I quote, "We exist as a vehicle to promote vibrant physical, emotional, and spiritual health in the individual." I think we're in for a special session. Please welcome Doug Kaufmann and Dr. Christine Salter.
Thank you so much. I need to tell you, I have learned so much from all of you. That was a hard act to follow. When I came back from Vietnam in 1971, I was sick, and all the king's horses, all the king's drugs couldn't help me. I knew I needed help. I probably saw some things a 20-year-old kid shouldn't have seen, especially as a corpsman. And I set out to try and help myself. It was a fascinating couple of years. I was, at that time, 21 years old. So what does a 21-year-old do when he comes home from Vietnam? He drinks and drinks and drinks, and he couldn't figure out, what adverse effect that would have. Monday, I was with a physician that I worked with, a pulmonologist. The police chief in a big city out in Dallas, where I'm from, had dramatic results from what this doctor did. He did a bronchoscopy on this guy. He's a pulmonologist, internal medicine specialist, and he found not a lung cancer, but an aspergilloma, a ball of fungus in his lung. And the doctor said, "Thank you for teaching me what you did, Doug." What I've been doing the past 45 years is trying and hoping to educate doctors on a couple of things. Mercury amalgam has been one. I've had all of mine pulled many, many years ago by a doctor out of Baylor and felt more improvement than I'd felt with everything else. So collectively, thank you all. I'm humbled, and I'm here to learn also. So fungi do parasitize human hosts. Dr. Everett Hughes, I took a job at USC Medical School when I got back, and literally I was paranoid. This wasn't the Doug Kaufmann that went to Vietnam. I couldn't ride the elevator up. We were on the 9th floor. I'd sweat, my mouth would be dry, my heart would palpate. And Dr. Hughes knew what was going on with me, and he said, you were in a land far, far away. Maybe you got into a parasite. And so this journey continues to this day. We didn't have Google searches in 1971. We had library cards. So I got a library card and went over to the university, and each night after I worked, I'd look into parasites. And I got this old 1953 book called Parasitology, opened it up. I didn't actually open it. Something opened the book to one of the chapters that said yeast. We if you guys, any of you were in Vietnam, we were covered with yeast from monsoons. We got new socks and underwear every 3 months. When we'd pull the old ones off, our skin would come off with them. And so I recognized I had horrible jungle rot at one time and a yeast problem. Well, this opened my eyes. You mean yeast can parasitize? Somehow it got inside my body. We didn't know transdermal back then, but this stuff got in my body. And that was an eye-opener. And then you looked at my diet, which just fed yeast or was yeast. You know, I remember Dr. Hughes saying to me, "What do you eat, Doug?" And I said, "Well, you know, Oreo cookies, bologna sandwiches, Coors beer." And he said, "Okay, you've got to quit that." 2 weeks later, I came back still paranoid, and he said, "Did you eat something?" No, I've switched over to Budweiser beer and chocolate chip cookies. This is the way. This is the way patients think, you know, and we just have to understand. I was one. So today we think there's about a couple million species of fungi. We've categorized 75,000 of those, and of those, only 300 are found to be pathogenic to man. Mind you, of those 300, they make poisons. Fungus, unlike bacteria, off gases of poison, yes, inside your body. I didn't know this at the time. They manipulate their host. They alter our immune system in favor of its own survival. They alter the behavior of their host. So diet is behavioral. You talk to a person who's got a bad systemic mycosis, a bloodstream fungal infection. I'd like to tell you they're rare, but we're finding they're not. And they're just like me, you know, 40, 50 years ago. They eat to satisfy not themselves, but these fungi. And look at the next one. In a symbiotic relationship, fungi became the dominant partners. We are studying yeast cells in every laboratory in the world today because they are so similar to human cells. Not bacteria, not protozoa, but yeast cells. Mycotoxins are secondary metabolites produced by these fungi and are capable of causing disease and death in humans and animals. This was quite an honor. In 2004, the doctor who founded the PSA test, Dr. Richard Ablin, immunologist in Arizona, I get to interview the most fascinating people on these TV shows. Hal Huggins was one of my favorites. I just absolutely love that guy. And Dick Ablin, you know, he was one of the senior staffers here on Oncology News. He heard my lecture and it blew him away, and he said, "You've got to publish this." And so I finally did in 2014. A couple of my doctor friends and I published this. These fungal mycotoxins we need to expound on. About 33 are known mutagens. They can mutate our DNA. Exposure ill effects range from annoying to life-threatening. They're capable of causing disease and death in humans. And then, according to microbiological reviews, frank growth of fungi on animal hosts produces the mycoses. Then we talk about mycotoxicoses. And basically, I just want to go over this with you because I think it's important. It is inhumane to study cancer with human subjects. So we use bunnies or rats. And they were doing that when I was there at USC Medical School. As a matter of fact, one day everybody was out to lunch and the UPS guy came to the medical school and I ran down the 9 floors of steps. And he had me check in 4 boxes. And these boxes were moving and they were squeaking. I'm in California. Liberate them. You know, boom, you're free, go, run. But I dropped these off on the floor where they study these things. How do you give mice and rats and bunnies cancer? You inject a mycotoxin, a fungal mycotoxin into them called Aflatoxin B1, and after a year they all have cancer, so you can study them. You want to study diabetes? You inject them with a different mycotoxin called streptozotocin. After 8 months, they all have diabetes, so you can study them. Has not one PhD gone home and laid down at night with his wife, put his arm around her and said, "You're not going to believe this. I just gave 90 bunnies cancer." How'd you do that? I gave them a mold. Are we getting into that mold? You know where I'm going with this. Most are immunosuppressive. More than 50% of these are immunosuppressive. All are heat stable. You can cook the rice, you can boil the pasta, but if they have deposited their mycotoxins within that food, you're eating it. Now look at this. This is fascinating. This hot off the press. FDA okays the first duodenoscope with disposable cap. After jury award $6.6 million in a Seattle cancer death. The germs from one patient were transferred to another, and this man died. Houston, we have problems. Remember the TB time test that the nurses gave us? They'd heat up the needle and then inject it into the guy behind us and all of us. You can't burn off mycotoxins. Autoclaving doesn't rid mycotoxins. This is kind of interesting in that I can find this reference absolutely nowhere on the internet. It isn't there. And so I had to go back and find one of my old books, and there it is. The American Cancer Society defines fungal mycotoxins as genotoxic carcinogens. We'll get into that a little bit here. Fungal mycotoxins can be, and just look at this, there isn't a tissue in our body. A tremendous book that was written 21 years ago stated that fungi can grow in any human tissue except the teeth. And what you guys are teaching me this weekend is, oh yeah, they can. Your teeth are always, we all have bloody gums and bones under the teeth and so forth. So these mycotoxins can get into any tissue in the body. Again, most are immunosuppressive. This is a very respected organization. It's called IARC. The International Academy for Research on Cancer. 5 fungal mycotoxins are listed as possible carcinogens. And fumonisin and ochratoxin, as you see at the bottom, cause cancer in laboratory animals. By the way, we found only one fungal mycotoxin is a known human carcinogen. It's called aflatoxin. This was discovered, and we learned it was a carcinogenic molecule in 1983. We don't want Houston, we have a huge problem. Other researchers say there are 7 or 8 that are now known carcinogens. But I'm going to lean on this one, aflatoxin, simply for this reason. It's in your corn supply. It's in your peanut supply. Varying amounts. I don't want to make anyone paranoid, but when you take a history on a cancer patient, which I've done thousands of times through the years, there's this craving of peanut butter. There's this craving of sugar that feeds the fungus once it's on board. So fungus makes lumps inside our body. You've heard of fungal polyps. Very often in the colon or the sinuses, we pull out these fungal balls and can stain them, and sure enough, they're fungus. What we don't hear much about is a mycetoma or an ascomycete. So let's learn a little bit about those. Most fungi produce microscopic spores inside special elongated cells or sacs. These are called ascomycete. And then a mycetoma clinically masquerades as a squamous cell carcinoma. Knowing this for so many years as I have, it just boggles my mind when people tell me, well, I have a simple basal cell, or I have, a cancer. Very often, if you put a little oregano on these topical things, they dissipate quite quickly. I had the honor of working for a guy named David Weekley, Johns Hopkins schooled dermatologist, who moved my family and I 30 years ago now from Los Angeles to Texas on a 5-year contract. He had a patient population that was huge, and he was on a special board for psoriasis. And this is important. He asked me if I would come in and help him with these psoriatic patients. He's got 6 PUVA boxes where people take their clothes off and stand there naked, get some UV radiation, get a cortisone shot, and go home, and they're good to go for a week. We did blood work on these patients and found a large percentage of them had mycoses. And so what we did, I just put people on a diet I developed, an antifungal diet, and he gave them diflucan, and those who had stomach problems, nystatin, and then we followed them for a couple of weeks. Dr. Weekley ordered his nurses to put Kleenex boxes in every room. 2 weeks later, just like this gentleman just said, 2 weeks later, the patients are emoting. It was absolutely amazing. Their skin for the first time was clear. They hadn't taken a PUVA box. We followed them for weeks and weeks and weeks, and it was one of the most rewarding things I've ever done. But in helping people with skin problems, you soon learn of their gut problems, and then their depression, and then these bumps on their arms. So these sac fungi, about 75% of all fungi are ascomycete fungi. The spores reproduce in this tightly woven sac where meiosis, mitosis, and nuclear fusion takes place. That's key. Major crops affected are corn, peanuts, and cotton. What is toxic shock syndrome? Sac fungi include cordyceps, blastomyces, histoplasma, cryptococcus, aspergillus, brewer's baker's yeast, and penicillium. Their spores can survive intact for indefinite periods of time in a sac-like structure, dimorphic. At body temp, they begin to change form from mold to parasitic yeast to invade and reside in the body. Dr. Weekly, when he died 5 years into our relationship, my wife and I and the kids were ready to go back to California, his patients, if you can believe this, opened a clinic for us. He had some prominent patients, and they funded a clinic so I would stay in Dallas. His wife gave me, upon his death, an old book. It was called Clinical and Immunologic Aspects of Fungal Diseases, circa 1957. Here are 4 or 5 different fungi that they said in medical school in 1957 mimicked cancer. Different fungi. Fungal infections can mimic cancer. This is kind of interesting. Fungal infections, including these, can mimic both the clinical and radiological findings of lung cancer. I heard this yesterday or day before. You're going to hear it here after I'm done. I think cancer is being grossly overdiagnosed in America. Fungal infections mimic cancer. This is fascinating, out of the journal Lung. Case series, 27 patients diagnosed both clinically and radiologically with primary or secondary lung cancer. They all proved to be fungal infections. These people got better on antifungals. We're missing something. We're categorizing everything as cancer. And it would amaze you after so many decades in this field how many people I've seen who, both of my sisters have had cancer. One of them 40 years ago, they're okay. They have to watch their diet, but they're okay. Okay, you've seen that one. This is fascinating, that it can be toxic to so many of our organs. Aflatoxin we need to talk about, one of the most potent carcinogens known to man, and has been linked to a wide variety of human health problems. The FDA established maximum allowable levels, but you know how that goes. So it's the only one of all the mycotoxins known to induce cancer in animals or humans. Aflatoxin is the only one tested. And I spoke one time, I interviewed one of the guys who did the testing on the trains, right? These big trains come in and they scoop out a little glass tube out of the train boxes and they shake it up and they go in and they do their laboratory work. Then they go to lunch, then they go to the bathroom, then they come back and they say, whoa, that's got aflatoxin in it. What's got aflatoxin, Doc? The train that was just here. Oh, that's already gone. That train is long gone. It isn't the germ. It's our immune system's ability to fight it. I mean, this is what I've learned in all of my years. So we inoculate study animals with this aflatoxin, and none of these researchers, just the most brilliant guys I've ever met, but none of them seem to get it. We didn't inoculate them with cancer. We inoculated them with fungus, and they began bleeding. Do you know how all of this started? Do you remember President Kennedy? I do. You know, I mean, I'm that old. President Kennedy's in office in 1960. 1962 in England, there are thousands and tens of thousands of turkeys dying. What they do is hire veterinary pathologists to come in and do autopsies on all these birds. Okay, this 7,000 died of bleeding. They bled to death. This 7,000, look at those lumps on their neck, and look when we do the autopsy, look inside, look at their liver, it's horrible. Nobody said, don't check the birds, check the food. One wise man came up and said, you know what's similar? For the past 6 months, I've been feeding all of them this. Boom, a 50-gallon drum of peanut meal. They checked the peanut meal and found something called aflatoxin. It's a mold that grows It's a mycotoxin that grows from Aspergillus mold, and we think some other molds. But we're commonly exposed to this, I think. It's cool to walk into a store, being me now, because every once in a while you see aflatoxin-free peanut butter. Kind of good, you know. Maybe I've made a little dent. So we know that we're exposed to this through eating it. This is fascinating because of this. This woman, Migdalia Arnon, pathologist in her 80s, still living in Florida. I do a television show that goes, you know, to 50, 60 million households. I get calls from doctors. She was one of them. Being a pathologist, I thought, oh boy, she's going to get on me about my cancer hypotheses. Let's go back. Fungus glows green. Cancer cells glow green. Ultraviolet lamps are used, and they break the seed of wheat and some of these grains that they're testing. And they use a fluorescence. They look, and if there's a green color or a green-yellow fluorescent, then they throw that grain away. It may contain aflatoxin. And there's the mold, Aspergillus flavus, and aflatoxin is the poison that it makes. So Migdalia Arnon calls me one day and says, hey, Doug, you are dead on. I said, well, thank you. And she said, I've written a book some time ago, 1976, and she said, as a pathologist, I would shut the lights off and I would get my, you know, little ultraviolet light. Cancer cells from these tissues all glowed green granules, all made green granules. So I see a parallel there that is absolutely amazing. All her controls were negative. Okay, so this question always comes up when I speak. Wait a minute, God gave us P53, right, the Superman gene, and God gave us phagocytosis that was just mentioned. What happens? How does this fungus get on board and how does it attack us? The most studied of all genes is P53 because damage to it allows DNA damage to proliferate. So says the American Cancer Society a long time ago. We humans all have P53 genes. By the way, it's identified, I think they're up to 55, 60% of all cancers have the mutation of our Superman gene that's supposed to protect us. Many examples of tumor suppression gene mutations, more are being discovered. What causes the P53 gene to mutate? So there you have it. One thing is the same thing we're injecting into these bunnies and mice and rabbits. Aflatoxin B1, made by Aspergillus fungus, is known to mutate P53 genes. And then you get deeper than that. Some of these tumor markers that you guys, you know, check routinely on your patients. 19-9, for example. If I see a 19-9, okay, lung cancer, you know, or pancreatic cancer, but ulcerative colitis is also determined with the 19-9, and then fungal infections. And the fungus it detects makes a sac. A cancer tumor or a sac fungus. Then you have the C-Myc, the NRAS, and so forth. These mutations were also seen in high frequency in animals with liver cancer that we injected the animal with, the mycotoxin, aflatoxin B1. So there's got to be confusion. We tend to be robotic in cancer work. There's got to be confusion. Well, your C-Myc is elevated. Look, your problem's getting worse. Or are you feeding the problem? Normally, phagocytosis, normally pathogenic yeast or fungi would be recognized as foreign, right? That's how our white blood cells work. And I went to half-price books and found this book for $1, Mechanisms of Microbial Diseases, circa 1989. Sometimes phagocytosis fails. Cryptococcus, an ascomycete, a sac fungus, escapes phagocytosis because the spores are surrounded by thick, viscous capsule. Gee, would that be the ascomycete? And then another, histoplasma. This is so overlooked. This is the first, I've spoken so many times, this is the first time I've ever heard anyone address bird droppings, histoplasma, cryptococcus, etc. But phagocytosis doesn't work. And I think it's because, I'll show you in a minute, Fungal DNA probes can isolate tissue. I don't know if you're familiar with these probes. There's a laboratory in Dallas that is doing these, studying these. They haven't gotten their FDA approval yet. You know what I'm talking about. But they are doing these. Soon we will be able to send an old biopsy. We'll be able to scrape an arm like a KOH mount that we now do for yeast. We'll be able to send it off to a laboratory and they can tell us not only, yeah, that's fungus or yeast, but they can analyze it quantifiably. And that's exciting. That's where we're moving. Okay, if not fungus, what do the pros say causes cancer? Heredity, gene mutations, or gene fusion? About 5 to 10% of cancers are thought to be hereditary, but back when I started in this, everybody's breast cancer, everybody's brain cancer was because mom or dad had it. Gene mutations, all cancers are thought to be caused by gene mutations, and once again, at least 33 mycotoxins are known to cause gene mutations. And finally, this one that I am interested in, genetic fusion. A hybrid gene is created by joining portions of 2 different genes, okay? Eukaryotes. Eukaryotes are human and fungal cells. Prokaryotes are bacteria, viri, and other cells. Many viruses, the Epstein-Barr, human papilloma, and you folks probably know this, hepatitis B and C promote human cancers by integrating their DNA into human cells. So hypothetically, it seems more plausible that if 2 eukaryotic, human and fungal cells would share the DNA, and less likely that a virus, a prokaryote, and a eukaryote would. It's therefore possible that integrated fungal DNA can also be found in human cancer cells, if only they were looking. Metastatic tumor is a hybrid of cancer cells and white blood cells. This blew me away. I spoke with Dr. Lazava. We became email pals. I spoke with her boss because they're on to something at Yale. Duke is doing very much the same work. This is exciting. Here was the headline. Our results provide the first proof in humans that metastasis can occur when a white blood cell and a cancer cell fuse and form a genetic hybrid. In the 1990s, I drew a little model. It's kind of like the flux capacitor. I'll show it to you in a minute. Did you know that the toenail fungus drug Sporanox inhibits cancer from metastasizing? Too many coinkydinkies here to not think this way. Both cancer tumors and ascomycete fungi grow in sac formations. Milton White, every one of you would have fallen in love with this man. He was in his 80s. I flew to Michigan to see him. His wife had just died. He was an important guy at the hospital. As he tells me this, he begins weeping. As Ruth, his wife, was dying, she grabbed his arm and said, "Milty, you're a smart man. You can figure out cancer." He died a few years later, but what an honor to meet his family and